I'm Charles Evans, so I get the privilege of talking to you guys today about hardware, something near and dear to my heart as an analytical chemist, and techniques that we would use in Metabolomics core. So just look at sort of the outline. You've seen this slide a couple times already. Hardware and techniques, you may see machines and techniques. This is a picky analytical chemist thing. We like to call our instruments rather than machines, but they are machines. So whatever terms you want to use are fine. This is where we fit in, sort of at the middle of a metabolomics experiment, and maybe some of the things that you see the least of in terms of your actual interaction with the core lab. You can decide how interested you are in the instrumentation. It's helpful to have at least a basic background, and that's what the purpose of this talk is, so that you can understand why we produce data the way that we do, and what some of the caveats and what some of the strengths of the methods are. So looking at this in a sort of a more simplified view, I wanted to take a moment to step back and clarify the difference between what you might think of what you do and what we do, and then the overlap between them. So if we just look at a biological experiment, which is broken up into several parts on the previous slide, sample preparation, which Lee talked about, instrumental analysis, and then the data analysis interpretation, which is itself many components. The classical division of labor in the core would be that you are responsible for the biological experiment with, of course, as has been emphasized many times, the recommendation that you consult with us in developing your biological experiment. But this is classically what you think of that you would do. Sample preparation mostly is transitioned over to us. You're going to submit the samples. There may be some aspects of sample preparation that you're responsible for. It may be something that's done entirely by the core lab. We certainly want you to know what happens to your samples after you get them and have input, so that if you know something about the way your samples should be handled, that we make the right decisions. Instrumental analysis is typically more or less entirely managed by the core lab. So what I'm going to spend a lot of time talking about is not necessarily something that you have to worry about unless you want to. And to the extent that you should be at least basically familiar with instrumental methods and techniques, so that you have an understanding of why we do what we do. And then data analysis and interpretation is where we transition back from what we do as a core lab to what you do. And again, there's many resources here that will help you in this process. And so we would really like to think of it as all a combined sort of working together, you and us. But this helps to keep it and clarify. So what we'll do today, you notice instrumental analysis, at the risk of being corny, is a black box. We're going to look inside that black box for a little while today and see what's inside. If you want to shut it at the end of this presentation, that's fine. If you want more information, we're here to let you know. Techniques, on the other hand, really affect every step of this process, of this workflow. So we want to let you know what the techniques are available, so that when you design your experiment, and you can use them, and then wherever you fall along the workflow, you can understand how it affects our analysis and the results that you get. Okay, quick outline. Hardware or instrumentation, we'll talk about, we'll do an introduction to metabolomics hardware, what tools are needed. We'll look specifically at chromatography and mass spectrometry in a little bit more detail. And then we'll consider why you would want to put those two things together, why a hyphenated technique. Okay, and then we'll talk about techniques. So if that puts you to sleep, or if you don't care, you'll probably want to wake up for this part of the talk, where we talk about the strategies that you'll use. Sue Penarthur has already told you a good deal about targeted versus untargeted. We'll review a little bit of that. We'll look at a workflow in some degree of detail. We'll look at an example of metabolomics data, how we go from the raw output of an instrument to useful information. Tomorrow we'll tell you, this afternoon, we'll tell you more details on that. We'll just look at from the lowest level raw signal to the beginnings of useful information. We'll take it a lot further. And then we'll spend some time talking about fluxomics, which is a specific technique that some of you may be interested in. You may find yourself interested in a future date. And it's different from the sort of obvious try to measure the concentrations of as many metabolites as possible. So we'll spend some time talking about that. Okay, hardware. So what tools do we need for metabolomics? Well, as Sue already told you, The idea behind metabolomics is to study all, or as close to all as you're interested in, or as we're able to do, small molecule, typically organic metabolites present in a cell, tissue, or organism, biofluid, any matrix that you're interested in studying. So how are we going to tackle that challenge? Well, we need an analytical method which is sensitive, because sometimes metabolites that we're interested in are present at very low concentrations. We need something that's quantitative. If you can't measure how much you've got, it's of very little to no value if you can say, yes, it's there, no, it's not there. So we need it to be quantitative. We need it to be universal. We need it to respond to a wide range of different compounds, the most polar, the most hydrophobic lipid-like compound, regardless of whether it's charged or uncharged. We need something that can do it all. And no method is perfect, but we want something that's as close as possible. And we want it to be able to detect as many compounds simultaneously as we can. If you wanted to measure one compound, for most compounds, there's nothing stopping you from going to Sigma Aldrich and buying a kit to measure lactate. We want something that can do a better job at looking at many things simultaneously to give you a broader profile view, whether that be all of a particular class of metabolite, like amino acids, or whether it be all of everything we can possibly measure if you don't really know where to start, or you want as much information to frame your future more directed studies. So what can do that for us? Well, the instruments that we'll talk about are liquid chromatography, mass spec, gas chromatography, mass spec. You may hear referenced occasionally capillary electrophoresis mass spec. That's another technique that's occasionally used. It plays a lesser role. We won't cover it in today's presentation. We can talk about that if you're interested. And NMR. My talk won't cover NMR. That'll be the subject of the next presentation. There's certainly advantages and disadvantages of each method, one over another. The core will help you determine what's the best instrument to use, and if you're not interested, we will select it for you and produce data from that instrument. Okay, so let's talk chromatography. Chances are most of you have had at least some basic exposure to chromatography in your previous research experience, but maybe not. So just a sort of a definition, chromatography is a means of separating and analyzing the components of a mixture. Typically in metabolomics, we're dealing with a pretty complex mixture. There's no sample, no biological sample that contains just one compound that you're trying to measure, or you could probably, you'd have trouble coming up with such an example. So we have usually very complex mixtures that we deal with. And we want to take all of those components and separate them out so that we can address them individually and determine what they are and how much of them there is in your sample. And the way it works is it uses the differential affinity of those components, read metabolites, between a stationary phase and a mobile phase. And the stationary phase is something which is going to absorb and then ultimately release your metabolite of interest over the course of the analysis. And the mobile phase is something which flows through that stationary phase and provides the other option. So your metabolite can either be stuck to the stationary phase or moving with the mobile phase. So let's look at a visual example of that. Here's the components of a typical chromatographic analysis. You have your sample, what you want to separate. You have a column, which is a tubular container, typically. I'll show you some examples later today in the demos. And in that column, there is a stationary phase. It could be a coating on the walls of that column. It could be tiny particles packed inside that column with a chemical functionality that gives it some property. Say it's very hydrophobic, or it's very hydrophilic, or it has a particular charge. And then you have a mobile phase, which is some sort of gas or liquid that's flowing through this stationary phase at a steady, typically, rate. And what we want to do is use this to analyze our sample. So the first step is that we inject our sample from our vial onto the front of the column, and so the mobile phase flows through in one direction. And we have everything, to a first approximation, sticking to the stationary phase, at least initially. And the mobile phase continues, or it flows through the column. We may do several things to get stuff to start to come off of this column, or the chromatographic term would be elute. We might change the composition of the mobile phase. We might change the temperature of the column. We might change the flow rate under some less common circumstances. And as that happens, the differential affinity of these components of your sample start to show themselves. So In the first step, we might have one metabolite that moves quickly through the column and is nearly immediately detected by your detector. And we'll talk about detector later. And then that comes out the column as a peak. And then at that same point in time, your other metabolites have moved less far through the column. And we may continue to change the properties of the mobile phase so that, say, if we're dealing with a more hydrophobic column, metabolite, that eventually will come off the column and give a second peak in your detector signal. And then finally, we'll continue to change the mobile phase composition or the temperature, and we'll get our third peak. And so that's the way chromatography works in a very general sense. You can spend easily a semester-long course talking about this, and so we're going to stop right there. The output of chromatography is a chromatogram. You're probably going to encounter this, at least in passing, in dealing with metabolomics. Just basically to define terms, a chromatogram is a plot of retention time, or intensity versus retention time. And as I just showed you, we had an example where we separated 3 things and got 3 peaks. Real mixture is often going to be a lot more complicated. We have multiple peaks over the course of the retention times in this, and various different intensities. Some things are smaller, they give rise to smaller signals. Some things are bigger, they give rise to bigger signals. We may not have perfect separation or perfect resolution of all the components. So that's the output of chromatography. Okay, LC versus GC. Which one do we want to use? Well, we can help make that decision. We may, you may tell us amino acids, and we'll already know which is the best method to use. But if you are, as an analyst, faced with that choice, you can choose between LC or GC. LC separates molecules or metabolites in solution. It relies on interaction between the analyte and the stationary phase, as already described. And many different methods of separation, modes of separation, are possible. We'll talk about that in a future slide. GC separates molecules in the gas phase. So you have to make your metabolite or molecule of interest go typically from the liquid phase into the gas phase by heating it. Chemical interactions, again, happen between the analyte and the stationary phase. Typically, the carrier gas, which is analog to the mobile phase, is inert. It's not interacting that much. It just carries the gas phase metabolites that aren't retained by the column through. Elution order is typically determined mainly by boiling point or volatility. There's other factors that play in. Generally, as a chromatographer, we say if you could use, and it made no difference, either GC or LC. GC is usually simpler. It's often faster. It often gives higher resolution. So if you can choose GC, you often will. But that's a big caveat. GC doesn't work for all metabolites. LC is a little bit more universal. It often winds that LC, I would say, 60-40 LC over GC, depending on the class of analytes that you're tending to look at. Here's a slide for your reference on some of the figures of merit of GC or LC. Both can be high sensitivity. Both can be good for quantitation if they're carefully managed. LC is the only one that's going to work with non-volatile compounds. Both can work with volatile compounds. There's more matrix effects concerns. We won't get into that in gory detail in the LCMS. We just have to do our best job to manage those so that we get accurate quantitative data. There's more modes of separation available on LC. This has already been described in passing by a couple of our speakers today. But you may hear, oh, you're going to submit samples. We're going to do a GCMS analysis. We need to derivatize them to make them more amenable to GCMS analysis. So the idea here is we're taking a non-volatile metabolite, something that you couldn't put into the gas phase without it burning up. Most amino acids would be like that. If you tried to heat an amino acid to the point where it became a vapor, it would probably thermally degrade before you got it in the gas phase. But if we chemically modify it, with something that typically has hydrophobic properties, we use a silating agent. Frequently, this is an example of methyl tertiary butyl silyl trifluoroacetamide, which is a derivatizing reagent that will replace active hydrogens on, say, oxygens and nitrogens with this bulky but non-polar derivatizing group. And believe it or not, when you get your derivatized metabolite, this thing has a higher vapor pressure, easier to put in the gas phase, makes some metabolites that aren't volatile to begin with amenable to GCMS analysis. But it does make the process more complicated. So we'll do it if we need to for GCMS. Another thing to mention about liquid chromatography, I mentioned that there are lots of different modes of separation. We will choose or help you choose the best method for the analysis, but you may encounter these terms in terms of reading metabolomics papers. You may occasionally encounter normal phase liquid chromatography where your stationary phase is polar, your mobile phase is nonpolar. It typically works for organic solvent-soluble lipids, but you can't have any water present in your sample, so it's not frequently used for metabolomics. Reverse phase liquid chromatography, abbreviated RPLC, uses a nonpolar stationary phase, and it works best for nonpolar to semipolar metabolites in an aqueous or a mixed aqueous organic sort of matrix. So this is fairly commonly used in metabolomics. Hydrophilic interaction chromatography is also commonly used. It's fairly analogous to normal phase, except it allows addition of some water to your mobile phase and to your sample matrix. So it's more compatible with metabolomics, good for polar metabolites. There are other modes of liquid chromatography you may encounter in literature: ion exchange, anion or cation exchange, ion pairing, size exclusion, which separates metabolites or larger molecules based on their size. All of these things are things you may encounter in literature. We will pick, or at the least help you pick, which method is going to be the best for the classes of metabolites. Okay, mass spectrometry. So what does mass spectrometry involve? Mass spec is a very useful, versatile technique. Every metabolite you measure has a characteristic mass or molecular weight. So we can use that property to help distinguish between metabolites, relatively obviously, and that's the sort of the basis of mass spectrometry. But a key caveat is that mass spec detects any compound or metabolite that can form an ion. So what are we talking about? An ion. Most metabolites, as they exist in solution in the body or in cells, are neutral molecules or are solvated. They're not independent ions. So the process of mass spectrometry requires that we convert a neutral metabolite into an ion. And we do that by a process called ionization, not surprisingly. And there's a variety of different modes of ionization that a particular instrument may use to form ions. The reason for pointing that out, though, is that not absolutely everything will form an ion under every mode of ionization. So if you have a carbohydrate, a very neutral molecule, you may have trouble ionizing it by electrospray mass spectrometry without some further tricks, derivatization, alteration of the pH of the liquid. So that's the first step in mass spectrometry, is we convert our neutral metabolites to ionized metabolites. And we also want to put them in the gas phase. So we need to, if they're in solution, we need to evaporate any solvent. That's typically done as in GC, that's already done for you. All your metabolites are in the gas phase. In LC, we use heat and gas flow to do that. That's part of the ionization source, taken care of by the instrument. I'll talk a little bit about that. But then the next stage, the key stage, is we want to separate them by mass. We want to be able to resolve something that has a molecular weight of 191 Daltons from something that has a molecular weight of 193 Daltons. And we want to be as specific as possible. We want to get as accurate as a mass as possible, all other things being equal, to be able to distinguish something with with a similar molecular mass, but that is still different from, say, another metabolite. So we'll use any of a variety of strategies to separate our ions that we formed by mass. And then we want to detect those ions and read them out in a way that preserves the information, that we can tell what their masses are, and that gives us quantitation, so that we get more signal for a more concentrated ion and less signal for a less abundant ion. So MS is sensitive selective. It's a high resolution method, and it can help identify metabolites because, as I said, everything has a mass, and it's a very useful property to help identify. It can also sometimes provide structural information beyond just that mass. What does the structure of that metabolite look like if you don't know? It's useful for quantitation, but sources of interference have to be carefully managed, and that's our job as the core lab, is to help determine if there are any potential sources of interference, and there usually are, and then manage them, typically by careful calibration, by inclusion of internal standards, by quality control checks, to know what the magnitude of any sources of interference are, so that the data that you get out you can trust, and at least to what extent you can trust it. Would we expect to be able to detect a 5% change, a 50% change, a 200% change in metabolite levels? Okay, so the output, just like I did with chromatography, the output of mass spectrometry is called a mass spectrum. You may encounter this in reading the literature. Analogous to a chromatogram, it's a plot of intensity rather than retention time. We have a plot of mass, really more specifically mass to charge ratio. We form ions. Ions can have a single charge, which is the case with most metabolites, or it can have multiple charges. You can have a +2, a +3 ion. The output is actually in a mass spectrum is the mass to charge ratio, sometimes referred to as M/Z. So in a given compound, in a given mass spectrum, you may have multiple different peaks that come from multiple different metabolites, may come from a reference signal. So here we have a peak for a metabolite, and we have an internal reference that is always present in all of our mass spectra that are acquired on a time-of-flight instrument. We'll talk about that, which we use to calibrate to make sure that the mass that we measure is as accurate as possible. And we may have other metabolites here. So we get two modes of resolution. We get both separation by chromatography and by mass spectrometry. Okay, very briefly, forming an ion, you may run into the term electrospray ionization. This is what I would say is the dominant method for forming ions in LC-MS that's used today for metabolomics. You have your liquid containing your metabolites flowing from your column. This is after a separation has occurred. We'll talk about that in a second. And we need to evaporate the solvent and form ions. So we use a nebulizing gas, a heated nebulizing gas, typically along with a drying gas to help remove the solvent. We also apply an electrical potential, high voltage, basically, to this droplet. This is all done by the instrument, which imparts an abundance of charge on the liquid coming out of this capillary. And in the process through the process of adding this nebulizing gas and applying the voltage, droplets are formed. Those droplets reduce in size over many successive steps of desolvation and fission, and ultimately you are left with unsolvated gas phase ions. And we can produce ions from a wide range of things. They can be very small metabolites, amino acids, very small compounds, even larger things can ionize. We typically don't look at proteins, but proteomics works using mass spectrometry works by very much the same methods. And so ultimately we get gas phase ions, which are pulled into the mass spectrometer and then analyzed. Okay, well, how do we do that? How do we analyze things by their mass? This probably is maybe more technical than is of interest to most of you. But I find it interesting. You may encounter these different references to different types of mass analyzers. So we'll spend just a minute or two describing how we would go from a mixture of different ions of many different masses to separated by mass and being able to usefully detect them. So ions have to be manipulated under vacuum. You'll find that out this afternoon. We have vacuum pumps running in our instrument room all the time. After you form the ions, they need to be manipulated under vacuum. Many different types of mass analyzers exist. There's a lot of different solutions to this problem. Here are a list of common mass analyzers that you may run across in the literature. We have at least 2 to 3 of these types of instruments in our facility. Quadrupole time of flight, and other users have ion traps on campus that we use occasionally. So basically, they're all solving the same problem. They just go about it different ways. One that you may run into is a quadrupole. Your ions come in, and quadrupole acts like a mass filter. It allows you to pass a particular mass to charge ratio at a time by altering its trajectory in an electric field. And then you can scan the electrical potentials that you're applying to these 4 poles to allow different masses to pass at different times. If anyone's interested in this in more detail, I can describe it to the extent of my limited physics knowledge. Really, this, even to us analysts, is not something we have to interact with on a daily basis. But the output of this type of instrument is relatively low mass accuracy. We can resolve things that are 1 Dalton apart, but not much better than that. But it's good for quantitation. And it's good in that it can be used in parallel with other mass analyzers. A time-of-flight mass analyzer is actually, this is the sort of simplest to understand. You put a bunch of ions in, you allow them, accelerate them to the same kinetic energy, and you allow them to drift in a field-free region. And bigger things, because K equals 1/2 mv squared, bigger things move slower. And so the small ions hit your detector first. You have effectively a very accurate stopwatch in your detector. And you say, how long did it take for my ions to get from a pulse here at the beginning of the flight tube to the end? You count when they arrive, and then that information gives you the mass. Believe it or not, it can actually be a fairly highly mass-accurate instrument with appropriate calibration. It's a little more expensive as far as the equipment. We have all of those. We have various types of instrumentation, so you don't have to worry about that cost. It does require careful attention to calibration. We use both methods. Tandem mass spectrometry is something you're probably going to run into in descriptions of types of instruments and assays that are out there. Tandem mass spectrometry is the idea is that we're going to, instead of just taking ions that are produced and figuring out what their masses are, we're going to do multiple stages of mass spectrometry within a single instrument, within a single analysis. So as before, we're going to put ions into the instrument. We're going to say, for example, pick one particular ion of interest. Maybe we're really interested in identifying an ion with a molecular weight of 237 Daltons. So we select that ion, and then we expose it to some sort of a collision-induced dissociation process. It may be a gas in a collision cell. There's various other processes that can be used inside the instrument. And so that particular ion is selected by a first mass spectrometer. Here I'm giving an example of a tandem quadrupole. So we use a first mass filter to select that ion. We use a collision cell, which could be a quadrupole or a hexapole. It doesn't really matter. But it's where that ion is broken up into multiple fragment ions. And then we get a spectrum of different fragment ions out. And we can use that spectrum to help identify. We can say, well, we know that a particular type of functional group will fragment to give a mass of a particular characteristic product ion, and that's useful to identify the ion. We can also use it for quantitation. We can say we know that a particular known standard has a parent mass or a molecular ion mass of 191 Daltons, and it fragments to give a daughter ion of 111 Daltons, and that is useful information for improving the selectivity for that particular ion, removing possible other interferences that might have had a similar mass to the starting ion, and then we can use that for quantitation. So a common instrument that you may hear of is a tandem quadrupole or a triple quad. This takes advantage of this property to give enhanced selectivity and sensitivity, typically for targeted analyses. Usually, if we're doing tandem MS, it limits the number of metabolites that we can monitor simultaneously. There are some ways around that if you want to do unbiased work on a tandem MS instrument. But often this is used for quantitation of targeted metabolites. Okay, briefly, why? Okay, so let's put it all together. Why do we want to use a hyphenated technique? Why can't we just do LC or just do mass spec and call that good enough? Well, LC and GC are useful for separating complex mixtures, but they may not be enough to separate a complex mixture, like a sample of plasma. You probably won't fully resolve all of your compounds by LC. And also, with just the output of a chromatogram, it's difficult to identify what metabolite is what. Mass spec can determine the mass of metabolites, and it's useful for identification. But if you put the entire metabolome into a mass spectrometer all at once, you're not going to be able to distinguish what all of them are. So when we put them together, when we run LC coupled to mass spec, we can do a better job with that. And so this is just sort of a basic block diagram. I'll get the chance to show you this as well as Chunhai this afternoon in the demo. But in LCMS, we have chromatography, which consists of a pump, a column, a means of injecting the sample onto the column. Then we have an ion source where the ions are produced. So the outlet of the column is coupled to the inlet of the mass spectrometer. We have the mass analyzer. This is a time of flight mass analyzer. I'll show you one that looks exactly like this upstairs. And then we have a detector contained with instrument and does data collection. A GCMS looks very similar, not physically, but as far as a block diagram, we have chromatography. It's a column inside an oven. We use temperature programming in GC, as Chunhai will show you later this afternoon. We do ionization inside the mass spectrometer. We have a mass analyzer, a quadrupole typically, or a time of flight to separate ions by the mass, and then we have data collection. Okay, so that's hardware. So if that was not of interest, you may want to pay attention to the techniques, because this will affect how you design your experiments. This is a little bit of review from Sub's talk, and we'll just sort of put a little bit of a different slant on it. Metabolomics approaches. As has already been mentioned, there's two basic designs of an experiment. You can do targeted, which is, in terse terms, if you know what you're looking for. If you know you want to measure amino acids in plasma because it's been published before, and you want to reproduce previous results and extend or have a different biological application, then that's a good place to start. And you can consult with the core, and we'll either use an existing assay that we have up and running. We'll develop a new assay for you if need be. We can reproduce other targeted metabolomic assays that are out there in literature. Typically, targeted metabolomics are useful for tens, maybe low hundreds, maybe not, in terms of number of compounds at once. There's nothing that stops you from doing multiple targeted analyses on a single sample. You may say, I want acylcarnitines, amino acids, and TCA cycle metabolites on a given sample. And you may still have the best way to go be to do 3 separate targeted assays rather than to move to an unbiased approach. Or it may not be, and we'll help you decide that. In targeted, we get confident identities, which are based on authentic standards, and we can give you either absolute quantitation, micromolar, or relative to tissue mass, or any other mode of absolute quantitation, or we can do relative quantitation, and sometimes that's good enough. An example would be free fatty acid profiling of blood serum. Untargeted, sort of a crude way of thinking about it is, if you don't know what you're looking for, if you have an experimental design, you anticipate there may be effects on the metabolome, and you want an unbiased look, or I'm sorry, unbiased is the wrong term. If you want an overall look at what may be going on in your sample, either in and of itself as a final data output, or to help guide you in future experiments, what other targeted assays you may need to do, what sort of biomarker or potential biomarkers might be present in your sample to do more investigation on. It can look at, so if you don't know what you're looking for, or if you want to look at everything as a first pass in order to help you guide future experiments. It can work for up to thousands of compounds. Some of those are going to be confidently identified. Some of them are going to be known unknowns. Some of them may be complete unknowns, something that we've never seen before, although that's less common. And the output, as many have already told you, is relative quantitation. And the example would be a global study of metabolic response to a change in diet. Say you're doing a dietary intervention study, and you just want to know what happens in terms of metabolites, but you don't have, you're trying to generate hypotheses rather than test specific hypotheses, or your hypotheses are multifaceted, and the data that you want is a multiple, is best served by this sort of approach. So just to look at workflow, I think Sue already covered this in a little bit of detail. The first step of any metabolomics experiment, including targeted, is sample preparation. Lee's already covered this in very good detail. Solvent extraction, sample prep, basically getting your samples ready to be put on the instrument. Then we do instrumental analysis, which I touched briefly on, and we'll talk. We'll demonstrate for you in more detail later this afternoon by LCMS or GCMS or CEMS. Then we're going to get raw data out of that, and we'll go through a couple steps to get to the point where we do quantitation, typically involving peak integration. We'll use standards to do to determine to calibrate our data, calibrate the instrument, determine how much signal corresponds to what concentration of analyte, and then we'll do data interpretation using statistics or whatever else is necessary. In untargeted metabolomics, as has already been described in some level of detail, the sample preparation and instrumental analysis will be tailored to a more general analysis of the samples, but the basic idea is similar. Instead of looking for targeted features, and Tanu and Sasha will tell you a lot more about this process than we have time to cover right here, but instead of looking for specific things, we're going to look for everything that may be present in the sample. So we'll do feature detection. We'll try to find what features are in common between the samples so that we can quantitatively compare them. We may do recursive processing to help find as many features as possible and make sure that the data is quantitatively as reliable as possible. We'll do data analysis. Often that'll start with some sort of separation by multiscalar analyses, PCA. Others will tell you more about this. Feature identification is an important step. Some compounds may be identified by virtue of their inclusion in our metabolite libraries. Some maybe are known unknowns where we see it reproducibly and we can report relative changes. And then we need to do more work if it's of interest to identify them. Some things may be true unknowns, and if they're of interest, we can do true. We can work more to help identify those compounds, and then further data analysis, statistics, pathway mapping, et cetera. Others will tell you a lot more about this. Okay, so let's give an example. I like to think in practical terms. Tanu will also provide examples. But how do we go from what came out of the instrument, what I was showing you, the chromatogram, the mass spectrum, to useful data? So the example that I'm going to give here is a targeted study of acylcarnitines in blood serum, let's say during exercise. Acylcarnitines are a class of molecule that are used in energy transfer in mitochondria as well as various other functional roles. They can have a variety of different characteristics. They can be, so the carnitine group is the common head group of the carnitine. It can have either a short lipid chain or even no lipid chain, a short chain acyl group, or a long chain acyl group. So here's acetylcarnitine and palmitoyl carnitine. This acyl chain imparts different functional properties to the carnitine, but they're of interest in understanding metabolic processes that occur during consumption of energy, during determination of what fuel is being used for exercise, for example, some related to my research. So we're going to use, as an example, blood serum from resting and exercising rats. And we're going to look at the levels of these different acylcarnitines. So the analytical method we already touched on, that we're going to choose. What we have to select is sample preparation. In this case, we use protein precipitation by an organic solvent, which removes proteins while retaining the solubility of the polar and moderately nonpolar carnitine species. And then we'll include internal standards in that extraction solvent so that we know that through all the steps that we're going to carry the sample, if any matrix effects occur, if we have loss of any particular classes of acylcarnitines, we'll be able to quantify that, and we'll be able to use it as a quality control check. And then we'll use, as our detection method, we'll use tandem quadrupole mass spectrometry following a reverse phase LC separation. Okay, so this would be the raw output if you were just to look at what came out of the instrument without any further analysis for this experiment. And we have what's called a total ion chromatogram, which means all signal from all of the ions that the mass spectrometer was able to perform or to produce, and you get a plot of intensity versus retention time. So many of these peaks here are indeed acylcarnitines that we're interested in. And this was a tandem quadrupole instrument, so we had previously programmed the instrument to look specifically for the acylcarnitines. And so we have a relatively clean spectrum. We can say that a lot of these peaks are indeed acylcarnitines. If we had a different type of analysis where we were doing more unbiased approach and we were attempting to detect all the ions, then we might have a more complex chromatogram. But even so, this is moderately complex. We just have peaks. We don't know what they are yet. We have some that are incompletely resolved. So we need to do more work to go from this point of the data to actually useful information. So the first step is to generate extracted ion chromatograms. So this is more terminology that you may bump into. Extracted ion chromatograms are where you take the total signal and you say, I want to look at signal that only comes from a particular ion, say a mass to charge of 204, which happens to be the mass of acetylcarnitine. And then, I'm sorry, this is an error in my typing. This should be a different mass here. Different carnitines will have different masses, and will have different mass to charge ratios that appear at different retention times on the column. So you can see here we have specificity not only by the mass to charge ratio, but also by the retention time. And the next step following that is to do something to quantitate. So typically we look at the area under the curve, or the integrated peak area, as our final as our preliminary quantitative readout, and that's going to be what is used then to do the further quantitation steps. So we measure the peak area for all of our metabolites, and then we use quantitation by a reference to a calibration curve. So previously we've injected samples of known concentrations that encompass the range of concentrations that we expect to find in a biological sample. Not too different from when you do any other sort of an assay, like a glucose assay or a lactate assay. It's just that we have prepared it in advance for as many metabolites as we hope to measure. And we are using an internal standard to compensate for matrix effects that may alter the quantitation depending on what other not of interest metabolites are present in your sample. This is what this looks like for a targeted analysis. We have software which aids us in this process of generating calibration curve, ratioing the peak area of your metabolite to that of the internal standard, generating and then finding where your compound falls on this calibration curve in order to measure a concentration. So that's a very crude work for those of you who have done quantitation by other methods. This is probably not unfamiliar to you. The trick comes in selecting appropriate internal standards and validating the quantitation and the recovery before you begin your assay. And then the output comes in targeted metabolomics can be as simple as bar graphs showing changes under the different conditions. It can be heat maps, which are good at displaying a wider variety of changes. It can be principal component analysis for unbiased work. It can be pathway mapping. So others will tell you a lot more about this process, and so there's going to be a lot more on that in this afternoon and tomorrow's talk. Okay, so we're going to spend the last 10 or so minutes talking about fluxomics. Conventional metabolomics is in concept relatively straightforward. So the idea is you're trying to measure the concentration or the total amount of small molecule compounds in your sample. So that's not too different from what you might do if you were running a biological assay for glucose. Fluxomics, or attempting to measure metabolic flux, is something that's a lot more challenging to do by other methods. So just to sort of define what's going on, the rate of turnover of metabolites is an important metric that you may not be able to access just by looking at metabolite concentrations. So this is a sort of a growing field of interest. The use of metabolomics and metabolomics methods to help understand rates of turnover of metabolites, and that may give you additional information that you may not be able to access from just, as I mentioned, metabolite concentration. So let's look at that in terms of an analogy. What is metabolic flux? If you haven't thought about it, and this is a slide that I lifted from the web, so I acknowledge the contribution from this individual. Not my idea to use it as an analogy, but I think it's effective in a sort of a corny way of describing what flux is. So if you think of your metabolite concentrations or your metabolite pools as what we're able to measure with conventional metabolomics, those are the size of a body of water in a river. Say before and after a waterfall, and perhaps in the middle of the waterfall we have this little pool. That's representative of your metabolite concentrations, or total amount. The flux is the rate of flow into and out of these metabolite pools. And that's reflected by the rate of flow of water from upstream to downstream. And so you have source fluxes, something, if this is your metabolite pool of interest, you have metabolism is a very dynamic process, as you well know. You have one or maybe several potential metabolites that feed in to a metabolite pool, and all of those things may have separate source fluxes. And then you have the flow out of that pool into sink fluxes. And so the key thing to notice here is that the amount flowing into the pool by nature has to be the amount flowing out of the pool. Otherwise, the pool is growing or shrinking, which could be happening. The direction of flow in this case is determined by gravity. In metabolomics, it's a little bit more complicated. You can get flow in both directions, so the analogy doesn't hold up perfectly. But a key key thing is that the size of the pool is independent of the amount of flow into or out of the pool. You could have a tiny trickle in a very large intermediate pool, or you could have a very tiny pool, and you could have a huge flow into and out of it. And both of those things, the pool size and the rate of flow, are useful pieces of information. So how are we going to address that? How are we going to figure out what this flux is? So this has been alluded to by various other speakers. We'll go in a little bit more detail right now. So let's use the TCA cycle, sort of central carbon metabolism, something most everyone has encountered and is probably of interest to a large number of investigators. As an example, not as the only thing that you might be interested in, but as an example of where you might want to measure metabolic flux. So this is a picture of the TCA cycle. We have glucose as one possible starting substrate that goes through glycolysis, multiple steps to form pyruvate, and enters into the TCA cycle through acetyl-CoA. The TCA cycle is not simply a cycle. It does more than just turnarounds. It turns and turns. It provides substrates for other synthesis of metabolic intermediates. TCA cycle metabolites can be consumed and produced by a variety of processes. So it's not as simple as this, but we'll focus in on this. So how would we go about measuring rates of turnover of metabolites? Well, we have to do something in order to be able to see it, right? So going back to our previous, but all we're able to really measure is we can't physically measure flow of metabolites. We need something that will give us information about it. So going back to our previous analysis, you can think about it putting dye in the river and watching how long it takes for your pools to become colored. Well, here what we're going to do is we're going to use a stable isotope, which is effectively dyed in terms of mass spectrometry. We can distinguish the difference between conventionally, or the most abundant isotope of carbon in nature, which is carbon-12, and another stable isotope of carbon, which is carbon-13, by mass spectrometry. If we put labeled glucose into this sample, say labeled with carbon-13, and then put that, we exposed a cell to that, and then gave it some period of time to incorporate that labeled glucose into the metabolic intermediates, then we might see something like this happening. Okay, so the first step, glucose will form fully labeled pyruvate, which will form fully labeled acetyl-CoA, and so 2 carbons from acetyl-CoA become incorporated into citrate by condensation with unlabeled oxaloacetate in the first step of the TCA cycle to give you a 2-carbon labeled metabolite. So we can now detect how much of this 2-carbon labeled citrate is present relative to the other isotopes of carbon, and then that follows through the whole rest of the TCA cycle. If we gave the cells more time, then let's say, okay, this is what happened in the first turn of the TCA cycle. We have our 2 labeled carbons. If we gave them more time, kept incorporating more labeled acetyl-CoA, we would eventually see incorporation of 2 additional carbons into citrate. We would lose some carbons to carbon dioxide, which is what the TCA cycle does. We would get a more complex labeling pattern. We would get multiple different levels of labeling in our TCA cycle intermediate. And we can look at the pattern of labeling over time as a means of assessing rates of flux. So how do you do this? So that's a complicated question. And again, I would reemphasize the previous emphasis that has already been placed is if you're interested in the flux experiment, come to talk to us. We can tell you about the various different types of tracers that are available, the various different types of analyses that are available, and the various ins and outs of doing the experiment. And this is a rapidly growing and evolving field on the cutting edge of a lot of research. So what may be available right now may change 2 years from now. It's not a new concept, but certainly the tools that are available now are much greater than they were 10 years ago. So we can do a lot more than was possible in the past. So let's think about it in cell culture, probably the easiest way to do a flux experiment. There are other ways you could think about doing this in animals or in humans under much more limited and specific experiments, although it becomes much more complicated, much more challenging. And if you want to go down that road, that's going to require extensive consultation and may or may not be possible at the present time. But in cell culture, let's say we took unlabeled cultured cells and we exchanged or spiked into their media a substrate that they can metabolize containing a C13 tracer. Say 13C glucose, uniformly labeled, would be one option. You can use other tracers. You could use labeled lipids, labeled amino acids. A variety of things are possible. and then you allow those cells to be exposed to the labeled tracer for various periods of time, say 30 minutes and 120 minutes. Then you harvest the cells at various different points along time following exposure to this labeled tracer, and then we go through our essentially conventional metabolomics experiment, and we do LCMS and GCMS analysis. What do we get out of that? Okay, well, if we were to look just focusing in on one metabolite, let's look at fructose 1,6-bisphosphate, which is a glycolysis intermediate. This is what it might look like as far as a mass spectrum in cells that have not yet been exposed to any stable isotope tracer after 60 minutes. We get basically a single peak with a little bit of natural isotope abundance. That's one caveat. 13C is at about 1.1% abundance in nature, and when you have a multiple carbon-containing molecule, we have a little bit more abundance. So we see some other peaks, but essentially all of the metabolite is contained in a single mass. This is negative ion mode, so we lose a proton. So a 340 Dalton metabolite shows up at a mass of roughly 339 Daltons. After 60 minutes exposure to 13C glucose, we have a little bit of it left, but almost all of it has transitioned over to the 6-carbon labeled fructose 6-bisphosphate. So we have fully labeled 13C, fully 13C labeled fructose bisphosphate. If we looked at earlier time points, we might have seen that transition. We might see, and we might be able to assess how rapidly that pool is turning over. Let's look at citric acid, a TCA cycle intermediate. Well, at time 0, we have, again, a single peak effectively, aside from the natural abundance, of citric acid at the unlabeled mass. After 60 minutes exposure, we still see some of that unlabeled citrate, but we now see a distribution of different isotopes up to 197 Daltons, which would be fully labeled 6-carbon labeled citrate. So at various different time points, we can see various different levels of labeling, and this is what that looks like. So at 10 minutes, we see a substantial portion of remaining unlabeled metabolite. We see predominantly of the labeled metabolite that's been formed, we see predominantly 13C2, which if you go back to look at the TCA cycle makes sense because the first step in synthesis of citrate in the TCA cycle is incorporation of 2 carbons from acetyl-CoA. So it makes sense that at early time points, the labeled form of citrate that we have is predominantly 13C2. As more time passes, we see evidence of increasing turns of the TCA cycle, formation of multiple different forms of labeled citrate, and we see increasing abundance of these higher labeled forms, decreasing abundance as time passes of the unlabeled citrate and of your 13C2 citrate. So this measuring the difference between these time points, and then to take it a step further, comparing differences in time points between, say, a control sample and an experimental sample is how you can go about, to a first approximation, measuring metabolic flux and getting some idea. So it's complicated. One thing to say, I already alluded to natural isotopic abundance. Carbon-13 is present in nature about 1.1% of all carbon atoms in your body, and in every organic molecule are carbon-13. This will affect the natural isotope distribution. If you have a molecule that only contained a single carbon, you would expect its 13C1 peak, meaning a single carbon labeled, to be 1.1%. But if you have a carbon containing 20 carbons, for example, like nicotinamide adenine dinucleotide, you see a substantial probability that you're going to have at least one carbon-13 in the molecule. So this is the NAD after no exposure to any 13C substrate. We have a lot of 13C1. And then, after time, we do see incorporation in other forms, but this will confuse your interpretation of the data unless we do something about it. So the core lab will, for you, if you're doing a flux experiment, do natural isotope abundance correction. So this is the same data, except for clarity, shown now without the unlabeled carbon. So we have a lot of the 13C1. This is your natural abundance, and a bit of 13C2 in the data, even at time 0. After we use our computational algorithms to correct for this natural isotopic abundance, we see no labeling. At our time 0, so it's the natural isotopic abundance has been removed, and we can look at only the new labeling that is present in the data. So when you do a flux experiment, the data that you get out is going to be natural isotopic abundance corrected. Just to say that this is something that can be done. there is the possibility of, beyond doing these simple experiments comparing relative rates of flux, of looking at or attempting to actually quantitate absolute rates of flux. This is something that is computationally intensive and is only as good as the data that you put in and the assumptions that you make. Really, at this stage, there are research groups that are exclusively dedicated to figuring out what is the right way to do these types of experiments. So this is an evolving field. If it becomes necessary for you to measure absolute rates of flux, it's something that we may be able to put you in contact with resources. We may be able to help work with you to measure absolute rates of flux. But I want you to understand that this is a complicated process, and it's not something that may be routinely deliverable, that we'll be able to report that the rate of conversion from hexose phosphate to fructose bisphosphate is 0.7 moles per minute per gram of tissue. That would be the ultimate goal, but most of the data that we generate at this time is used for comparing relative rates of flux. But the idea behind this is you would measure as many metabolites as you can, define your input parameters, attempt to solve a model of the metabolic networks computationally, observe the output, refine the model, and then ultimately generate accepted flux data. So that brings me to the end of my talk. I know that's a lot of ground in an hour to cover, but we'll talk. So hopefully I showed that combining a separation technique like LC or GC and mass spec really does offer a pretty powerful and universal way of detecting metabolites. There are certainly caveats to look at, and we as a core lab do our best job to manage those and produce data that's useful to you, so that you don't have to worry about all of those things. But it's good for you to be aware of them. We use multiple instruments and try to pick the best analytical method to enable a comprehensive study of the metabolome or whatever portion of the metabolome that you're interested in. It can be done using either targeted or untargeted approaches, or often untargeted will inform a targeted experiment or the other way around. And fluxomics is the process of using stable isotopes. I should point, these are non-radioactive isotopes. A lot of times other biological experiments are being done with radioactive isotopes. This is the analog where we can get away with using non-radioactive isotopes. It opens up a lot of experiments that are not otherwise possible to study the rate of turnover of metabolite pools in biological samples. And that's all I've got. So I'm happy with what limited time remains to answer any questions.
The first slide you showed us for the fluxomics. Mm-hmm. Which part do we do and which part do we do?
Right. Great question. Great question. So you are going to be involved. So again, I will emphasize, especially for fluxomics, talk to us. That's right. Where that transition happens is going to be dependent on the particular experiment. Chances are, the first time you do this experiment, you're going to want to come to either get intensively familiar with the process by consulting with us and talking through it, or you're going to want to work with us in our lab to understand the right way to handle the cells, to give them the tracer. You're probably going to want to talk to us to see, we have limited quantities of these stable isotope tracers that we can give to you, and in small scale experiments they're not that expensive. For larger experiments, you'll want to know what to buy and where to go to buy them. But once you become familiar with the process, and we'll work with you with that, and you're just doing repeated experiments, you can manage up to this point right here. So you're going to expose them to your stable isotope labels. There's no hazards in working with these above any other media, so you don't have to worry about radioactivity. So you'll be able to manage up to this point, quenching metabolism in your cells and collecting the samples, and then you'll submit them to us. And we're going to take them and do the metabolomics. We're going to produce the data, and we're going to produce output, which will show you the relative extent of incorporation of isotopes into the experiment. And then we'll help you interpret what that means as far as rates of flux.
For a lot of these experiments, we've gotten to experience a lot of different cell types, both adherent and cell suspension. Actually, we've begun now some in vivo experiments, both in animals and humans. So we have an idea of what's going on. But I think you should know that you should expect to do some preliminary experiments just to see how fast metabolism is going on in your cells under specific conditions that you're going to choose. Because we don't want to say do a 60-minute experiment and you're at isotopic equilibrium all across, or you want to do a 5-minute experiment and you get very little label that we incorporated in. So unfortunately, some of this is going to be experiments, and that's why we call it experiments. But some of the cells, I think we have a pretty good idea where we're going to be able to give you a pretty good idea of what might work for some initial experiments to do it. And then it also depends upon what you're really trying to label. If you're trying to label glycolytic intermediates, seconds is probably long enough. 2 minutes is long enough. If you're trying to get label into purine pathways or things like that, it's going to take longer to label up. So this is why you really need to talk to us about and specific. We can get an idea from what you understand, specific metabolism in your cells, and what our experience. We should be able to help you do that. So Charles, so just because you know better than I do, so a lot of people want to know, so how much is to do let's say 20 dishes of that with glucose metabolism? How much is C13 glucose or cost?
Sure. So the cost of C13 glucose is actually pretty minimal. We can buy a gram of C13 glucose for just under $100. So when you're talking about 5 to 25 millimolar in cell culture experiments, and you're using uniformly labeled glucose, you don't worry about it. It'll be a very small cost relative to your experiment. If you're thinking about doing other tracers, things more like lipids, so I mean, yeah, you're going to talk a few dollars worth of isotope for that sort of an experiment. That's all. If you're thinking about tracers like glutamine, you can think tens to low hundreds of dollars for experiment. If you're thinking about doing this in animals or in humans, where you need grams and grams of this stuff, the costs go up substantially. So if you're talking about trials of exercise on a bike where you're infusing a stable isotope tracer, it's a substantial cost and it needs to be budgeted into your experiment from the beginning. First, come to talk to us. There's people who can help with those sort of experiments. You may or may not even want to go down that road. But for cell culture experiments, it's not a zero cost, but it's not untenable for most tracers you're going to want to use.